So yeah, my supervisor is Tim Saunders, who has an experimental lab in the Center of Mechanobiology in Warwick Medical School. So a lot of my projects are very specific to some biological problems, which I'll try to make the motivations clear today, which I have the time for, so that's good. So I'll be presenting on in-silico image-based modeling of morphogen diffusion. So first of all, what are morphogens? Why are they important and why we care about them? So during embryonic development, all organisms start off with stem cells, which are pluripotent, which means that they have the ability or the potential to become more specialized cells. So for example, all of us, we have liver cells, cardiac cells, neurons, and these cells are all phenotypically different, and they have undergone a process called cell differentiation. So to become all these specialized cells. And the process of cell differentiation is nicely illustrated using this Waddington's landscape model. So imagine a cell or a cell fate is like a ball rolling down the hill. And when they roll down the hill, it's like a process of differentiation, and they'll enter these valleys or attractor regions, which represent cell fates. So you see the intermediate, there's this intermediate valley, which is like an intermediate fate, which we call progenitor cells that have the potential to even further differentiate into even more specialized cells. And the bottom of the hill would be the most specialized adult cells that we have. So during embryonic development, cells in the tissue will need to know which hill to roll down, like how to differentiate. And so that information is basically encoded in morphogens. So these are proteins or signaling molecules that provide spatial information to cells in the tissue to direct these cell fate decisions to make your complex organs during morphogenesis. So how they give spatial information is by forming a gradient in concentration across the tissue. So typically, you have a source region for the morphogens, and this is a region of cells that will constantly produce these morphogens. And then you have a target region in the tissue, and these morphogens are transported across the tissue to form these concentration profiles, such as an exponential one shown here. So I'll give some examples from live systems. So this is a fly embryo, and we're looking at this bicoid morphogen, where you can, which concentration is indicated by this intensity of the image here. You can see this, right? So this gradient gives the polarity in the anterior-posterior axis. So the high concentration region in the anterior will eventually form the head of the fly embryo, and then the low concentration region will eventually form the tail end of the embryo, and then the middle part would be the body segments of the fly. So another example is the developing spinal cord. So a developing spinal cord is a long rod-like structure, and if you take a cross section in the developing spinal cord, it looks something like that. And this morphogen called Sonic Hedgehog, which is named after the video game Sonic the Hedgehog, gives polarity in the dorsal ventral axis of this spinal cord. And depending on the concentration of the Sonic Hedgehog in the regions, you will form different types of neurons. So the bottom here will be motor neuron, up here will be sensory neurons. Basically will form your complex central nervous system. And finally, related to this project is the Wind morphogen that is involved in neurogenesis of the zebrafish embryo, which is the development of the brain. And the concentration of Wind is indicated by the intensity of this green color. So the reason that we can see fluorescing in green is because in your experiment we have encoded that we have fused the gene that makes Wind3 with a gene that produces green fluorescent proteins, or GFP for short, which is a naturally fluorescent molecules. And so during your experiment, when your system produces Wind, you can see it fluorescing in green, and that's very helpful because we can indicate the regions of high concentration and low concentration of Wind in your experiment under the microscope, which is really handy for analysis. So as I mentioned before, these morphogens have to be transported across our tissue, but how these morphogens are being transported are still being studied and not very well known. And there are a lot of debates on the different types of mechanisms to transport these morphogens. So there are two main groups of mechanisms, cell-based and diffusion-based. For cell-based, I've listed here transcytosis and cytonemes. So these are mechanisms of transport that is guided manner, guided by molecular interactions. So cytonemes, for example, they act like train tracks across that transports morphogens from cell to cell. And it's a very kind of targeted mechanism. On the other hand, we have diffusion-based mechanism, which relies more on the random motion of particles. So traditionally, we assume that morphogens are freely diffusing in your tissue. But if the morphogens are localized in extracellular space, means outside the cell, and if the tissue is packed with cells, these morphogens will have to navigate through the tortuous extracellular environment to get from the source to target regions, and they might also encounter receptor bindings that will slow down their movement. And so that points towards this hindered diffusion model. Recently, with increased resolution in imaging techniques, studies using single molecule tracking experiments have shown that morphogens such as nodal and lefty moves in the extracellular space, and the mobility is reduced when they are transported across these tight extracellular spaces, and this is supporting the hindered diffusion model. So experimentally, how we measure diffusion coefficients of the particles, there are two experiments here, which is important for the rest of the talk. So I'll try to explain them. So firstly, we have fluorescence correlation spectroscopy, or FCS for short. So this is when we focus a laser beam on a very, very small area of our tissue. And so this tissue has, again, fluorescent molecules. And the region that we focus on is really small, such that there's only a few particles diffusing in the region at a time. And whenever a particle enters the region, they are fluorescent, and this is detected using a photon detector, and you detect that for all the time steps, and you get an intensity time series. Then you can analyze this time series using a temporal autocorrelation function and infer your diffusion coefficient. On the other hand, we have FRAC, which is short for fluorescence recovery after photobleaching. So this is where, again, you have a tissue which have fluorescent particles diffusing, and then you photo-bleach a region. This is when you give light exposure to the fluorescent molecules, and then they'll lose their ability to fluoresce. So the properties are still there, it's just that they've lost their fluorescent ability, and that's an irreversible process. Then we allow the system to recover, so they'll diffuse and they'll mix with the rest of the particles that are still fluorescent, so the intensity will recover. And looking at how the intensity recovers, you can infer the diffusion coefficient. So experimentally, these values have been found to differ massively. So for example, the wind morphogen, the zebrafish, is measured to be around 60 microns squared per second using FCS. And that's because we are measuring the local diffusion. Because the region is so small, we are measuring the local diffusion coefficient of the particles. Whereas using FRAC, it's measured to be only around 1 to 2 microns squared per second. This is because you are looking at a much larger area in the tissue, so you are looking at diffusion at the tissue scale and in the more global sense. And this difference in measurement is also seen in all different types of morphogens and different types of model systems. And this difference in measurements have led to confusion in the field, and is also used to argue that diffusion is not a viable transport mechanism for robust patterning and supporting other transport mechanisms that are the more cell-based ones. So motivated by this difference in measurements from the local and global diffusion coefficient, and also the single particle tracking experiments, we aim to build a in-silico model that utilizes realistic biological images to show that properties such as tissue architecture and receptor binding alone can reproduce this slowdown from local to global scale of diffusion coefficient. To do that, we first need images that are of high enough resolution. So I'm not going to go through this, but this is done by our collaborators in NUS Singapore. But this is not trivial, and it's actually been a disadvantage sort of in the past for these type of simulations. So using a combination of imaging techniques and 3D segmentation, we divide the cells up from our images and some image analysis, we can obtain high resolution of 3D representation of the zebrafish brain. And this is an example of a 2D slice, which is a binary image. So the white spaces are the extracellular space, or ECS, and the black spaces are occupied by cells. And from our zebrafish brain images, we can obtain images from different regions of the cells, different regions of the tissue. So even from the 2D binary slice, we can see that the cerebellum and the midbrain hindbrain boundary regions have very different structure to the optic factor regions. And this is just a representation of the 3D ECS skeleton that's made from the stack of these binary images. Okay, so now with the high resolution images, we can perform our particle models. So that's just the diffusion equation, and then we have the simulated diffusion coefficient for the particles. Then we simulate individual particle trajectories by just doing simple Brownian motion update rule, which where you sample from a Gaussian standard normal distribution, and the standard deviation would depend on your simulated diffusion coefficient. So I'll give an example. So we randomly simulate the particle trajectory, but then now we have to localize the particles in the ECS, so we just move them back into the ECS. And then we do that for all the time steps for all particles. The motion would look something like that, and it's all in the ECS. And our goal is to estimate the effective diffusion coefficient that will differ from the simulated parameter, because now the particles are being confined. So to do that, we simulate FRAP experiments, where now we photo bleach a region, a circular region in our tissue architecture. So this is a zoomed in version to the center of the architecture to see the particles. The black particles are the ones that are being photo bleached, and then the green are the fluorescent particles. We also assume instantaneous photo bleaching, and then so this is time 0. Then we just allow the particles to diffuse and recover over time. Yeah. And then we can obtain FRAP data by simply calculating the number of fluorescent particles inside the region for each time, and divided by the total number of particles to normalize the intensity. Yeah, then we get recovery curves that look like that. And then we can fit, so there's a analytical equation which we can fit for to get the diffusion time constant for our diffusion and estimate the effective diffusion coefficient from of the particles. And then we also perform FRAP experiment on all the different zebrafish brain regions that I showed before, and we also perform it on as a control on the no cells case, which means particles can just freely diffuse. And now we plot the effective diffusion coefficient that is estimated from FRAP against our simulated diffusion coefficient parameter. So the no cells case, as we would expect, would recover the simulated parameter, but with added tortuosity now in other tortuous environments, the effective diffusion coefficient is now reduced. This is quantified in this table here. So the middle column is the factor slowdown, which is basically 1 over the gradient of this straight lines. And the volume fraction here is the volume of the ECS to proportion of volume of ECS over the entire tissue volume. And this parameter has been shown in previous literature to be the main factor in determining the slowdown of the effective diffusion coefficient. So you can see that just with different structural differences, you can see that the cerebellum and the midbrain hindbrain boundary gives a larger slowdown than the optic factor. And next, we also perform in-silico FCS, where now we focus on a much smaller region, again in our 3D diffusion model. We just diffuse it and count the number of particles in the region for each time step, and we get this particle count time series. Then we perform autocorrelation on this time series, so that's the autocorrelation function. The angle brackets are just the temporal average, and the autocorrelation function, so something like that. So for the first graph, the different colors are the different are FCS performed in different regions of the zebrafish brain. For the second plot, it's different colors are the different diffusion coefficients. So a bit about the autocorrelation function. So the x-axis is the lag time, so tau, and this is plotted in a log scale. The G naught gives, because of how we normalize it, gives the concentration of particles, so 1 over the mean number of particle count in our region, in our time series. And the steepness of the autocorrelation gives, from there you can infer the diffusion coefficient. So the faster the diffusion coefficient is, the faster your autocorrelation curve will go to 0. And again, there's also a analytical function which we can fit to for an FCS autocorrelation. And we can infer our local diffusion coefficient. So now we can plot the local diffusion coefficient that's inferred from our FCS experiment. You can see that it pretty much recovers our simulated parameter, even if there's a tortuous environment. So this shows that at what scale you measure diffusion in matters, and the surrounding tortuosity will also affect the diffusion coefficient measurements. So one of the benefits of in-silico modeling, which you can't do in vivo, is that you can alter the in-silico environment pretty easily. So here we look at how the connectivity of the tissue architecture will affect our FRAP simulations. And this is done by adding dead ends to our 3D ECS channels, and different amounts of dead ends. And we also play around with the volume fraction parameter by dilating our images, so dilating the white spaces of our extracellular space, and performing again FRAP on the different tissue architecture that's altered. Now we see that focusing on this green box, we see that with added dead ends but maintaining or increasing the volume fraction actually further slows down the effective diffusion coefficient, meaning that volume fraction is not the only parameter that is used that affects the slowdown, but also the connectivity of the tissue architecture. So here we conclude that the zebrafish brain tissue architecture alone slows down diffusion coefficient by a factor of 2 to 3. But that is not enough, if you remember from the start of the talk, to account for the factor 50 to 60 slowdown of the diffusion coefficient. So then we consider receptor bindings. So this is the usual receptor binding reaction. So your free particles will react with free receptors to form a bounded complex that is relatively immobile. And we give some further assumptions to the receptor distribution being in equilibrium in all around the extracellular space, as well as cell surfaces, and they are in abundance compared to the number of free particles. Then we can reduce the reaction to basically this, where freely diffusing particles will become bounded with some rate small k on, and it's released with some rate k off. And we can vary this ratio k on over k off and see how our effective diffusion coefficient is affected. So these are the values with different parameters of k on over k off and simulated diffusion coefficient. Again, this is FRAP simulations done on a tortuous extracellular environment. Focusing on the diffusion coefficient 60, the decrease is sort of logarithmic trend with the parameter k on over k off. And now effective diffusion coefficient can be further decreased up to a factor of 50 to 60. And these values have been shown to be in line with the Wnt binding in zebrafish with their receptors HSPG and receptor and another receptor fizzle in vivo that is quantified in this paper shown here. So with that, to conclude, receptor bindings and tortuosity alone can reproduce this local to global diffusion coefficient slowdown that is seen in experiments. So the in vivo environment is extremely complex, and so experimentalists would need to handle FRAP data with extra care and not rule out sort of diffusion as a viable form of transport mechanism. And this is one of the benefits of in-silico modeling for biological systems as well, because we can very easily separate the different contributions and individually analyze them. And also we can tune the parameters in silico pretty easily. So we are not the only ones that have been looking at this. With recent imaging tools and also computational power, there's been a lot of kind of interest in simulating diffusion in sort of these complex environments. And some with the different methods. So this one, for example, I think they uses the continuum methods instead of particle modeling. And this is our preprint on the thing, on the research and the GitHub page for the particle model and also the in-silico the image data. And thank you to my lab for support and also our collaborators at NUS. Thank you for listening.